Okay, greetings. We're going to be focusing on now the special senses. Now, this chapter will review the 5 special senses: vision, hearing, balance, smell, taste. You have to keep in mind that all of these senses have much more complex structures, and this is set up so that they can basically do the special detection of the stimuli. Now, we're going to start off with olfaction. I would also encourage you to review the spotlight of olfaction on pages 568, 569. When you really talk about olfaction, that's a sense of smell, and it occurs via a paired or olfactory organs that are located in the nasal cavity. Now, this is on either side of the nasal septum, and it has a special arrangement of olfactory sensory neurons. Now, in the neurons, they have specialized receptors. But that olfactory dendrites, as they might be called, are basically coated with kind of a mucousy, watery mucousy material. When we define odorants, they are water-soluble and lipid-soluble substances that are detected by the olfactory sensory neurons. Sniffing, when we do this, it helps speed up the exposure of odorant molecules with the olfactory epithelium. You notice where the actual olfactory epithelium is. It's in the superiormost part of the nasal cavity. Now, the anatomy of the olfactory organs. Basically, you have this area called the olfactory epithelium that contains the sensory neurons. The interesting thing is that the sensory neurons have a high turnover, and so they're constantly going to be replaced from within a short period of time. They have supportive cells as well as basal epithelial cells, which act as stem cells. They will replace the dying olfactory neurons. On the extensions of these cells, you have these sensors, and the sensors respond to different parts of a chemical molecule. Not the whole chemical molecule, but different parts, and I'll get into that in a little bit more in a minute. The epithelium covers the inferior surface of the cribriform plate, the superior portion of the perpendicular plate, and the superior nasal conchae, and you can see that on the diagram. The underlying lamina, the lamina propria, consists of basically areolar tissue, blood vessels, and nerves. Also, that layer has olfactory glands that produce a thick pigmented mucus. This mucus is interesting because it also absorbs water and therefore keeps the olfactory dendrites epithelial area moist. Now, basically what we know is that the olfactory sensory neuron fibers pass through the cribriform plate and then project into the olfactory bulb at the base of the brain. This is really the first synaptic site, so you have certain processing that occurs in that bulb. But then the rest of the information passes down the olfactory tract, which happens to be cranial nerve number 1, and carrying the information to the olfactory cortex. Now, there is a cortex on each of the cerebral hemispheres. And then it, interestingly enough, will project fibers to the hypothalamus as well as to portions of the limbic system, which accounts for things like, wow, I have a certain feeling when I smell this thing, maybe disgust or pleasantness, maybe that I have a certain memory about a certain smell. It depends. Now, olfactory receptors, as I said, when you have the odorants bind to the receptors on the olfactory epithelium, you have to think about this. Since the dawn of humanity, since the dawn or the evolutionary development of man, we've had this interesting thing. How is it that we can detect now gasoline, burning plastic, and a variety of other chemicals, especially since we make approximately 1.5 million new chemicals each year? Many of them have unique odors that we are able to detect. And that's because the odorant molecules, there's not one for gasoline, not one for roses, not one for geraniums, not one for, moldy smelling. It's really that the detectors, or the receptors, are binding to different parts of the actual odorant molecule. A benzene group, a hydroxyl group on the chemical formula, basically a methyl group, or any of a variety of other parts. And that all of that information is summarized back in the olfactory bulb, then passed and further processed by the olfactory cortex. Now, what happens when you have the odorant bind to the receptor? The receptor activates a G protein complex, which of course then stimulates, ramps up the production of cyclic AMP, which then opens sodium ion channels in the plasma membrane. This will create generator potentials, and these depolarizations can trigger then an action potential. Now we come up to another issue, which is olfactory discrimination. I've talked a little bit about this before, the summation of the information from different components of the chemical compound. Olfactory receptors, though, are frequently being replaced, and I've talked about how the basal epithelial cells divide to form new sensory neurons. It is interesting to note that as we age, though, the total number of olfactory neurons will decline with age. Those that remain are less sensitive. This explains why you have situations where, like, many of the elderly have difficulty detecting odors and hence require more stimulus. For example, some people that wear a large amount of perfume, it is not that they just wish to or consciously are aware that they have an incredible amount of this odor, of this perfume, but really it's the higher amount that they have to put on before they're actually able to smell it. Okay. We move from there, olfaction, to gustation, taste. And when you talk about gustation, that's the sense of taste. We have gustatory epithelial cells, or taste receptors. They're found in taste buds that are distributed over the entire superior surface of the tongue. And you will find some distributed in the adjacent portions of the pharynx and larynx. These receptors are stimulated by dissolved substances, food molecules. The stimulation leads to action potentials, which are sent to the gustatory cortex for interpretation. There are actually 6 taste senses. Now, we're commonly aware of salty, sour, sweet, and bitter. But in the past 50 years or so, we have discovered umami, which is sort of the meaty, brothy taste, as well as a discovery of a water sensor. Now, we don't taste water in the sense of, it's this good water or whatever, but it detects this, and then we'll explain just a little bit more about how this affects and sends information back to the hypothalamus. So let's talk about the anatomy of the papillae and the taste buds. On the surface of the tongue, there are various epithelial projections. They're called lingual papillae, and they form distinct shapes. The filiform papillae, which is sort of fili for thread. The fungiform, which is sort of like a fungus or a mushroom papillae. The valate, which appears as sort of a wall-like structure. And then the foliate, which is sort of a leafy in appearance. And you can see these all in the diagram. The distribution varies by region, and the components vary, but most will contain taste buds. Now, if you take a look on the diagram also, the taste buds will contain about 40 to up to 100 gustatory epithelial cells. And they have many basal cells, which also, again, act like stem cells to replace those taste buds or taste cells that die out. There are a variety of ages or stages that the basal cells are in. The basal, the transitional, and the mature. And it's only the mature that are gustatory epithelial cells. These cells at all stages, though, are innervated by sensory neurons, and you can see that in the diagram there if you look closely. The interesting thing is what differentiates the mature cells from transitional, etc., is that the mature cells will have these microvilli. They're called taste hairs, on which you have the actual taste receptors. The taste hairs will extend from the taste pore, which is a narrow opening on the surface of the tongue. And that's where the chemicals will actually come in contact with the taste receptors. The lifespan of a typical gustatory epithelial cell is only about 10 days, which is why you have to have so many of these cells going through this various cycles and aging, because some will die out eventually and they're being replaced. And when we talk about the gustatory pathways, we have to understand that the taste buds are innervated by cranial nerves 7, 9, 10. The facial nerve innervates the anterior 2/3 of the taste receptors in the tongue up to the valate papillae line, which you can see that valate papillae line there on the diagram. The glossopharyngeal nerve innervates the posterior 1/3 and the valate line of the taste receptors. The sensory afferent fibers then travel to the solitary nucleus of the medulla, and then the postsynaptic neurons travel to the medial lemniscus in the midbrain, which send information to the thalamus. Now, I want you to be mindful of one other thing. Many people would state, yes, this has happened to me, that when I have a cold and my head's all stuffed up, I really don't taste food well. Well, this is the concept of flavor. Flavor is really a combination of the senses of both taste and olfaction. Without olfaction, you get the basic taste of, sweet and sour, etc. But without either, the basic sense of taste is merely not even there. You basically have something, but it's not really much in there. Because more or less you have to keep in mind that when we talk about what food manufacturers talk about is a mouthfeel, which includes taste. But it includes, the aroma that's picked up, of course, by the olfaction, and then also the mouthfeel in the sense of, is it smooth, is it creamy, is it gritty, is it this and that? So we're talking about other mechanoreceptors that are in the mouth. They don't play a role in taste, but they play a role in sort of pressure and other tactile sensations. Now, I went to gustatory discrimination, and you can see this in 17.2b. As I said, there were 5 primary tastes: sweet, sour, salty, bitter, umami. Sweet, the receptors are linked to G proteins called gustoductins, which trigger secondary messengers inside the cell. This also occurs for bitter. Salty and sour respond to different, basically, presence of salt solutions for salty, acid solutions, which is basically the trigger is hydrogen ions, for sour. Both of them have sodium ion channels that lead to the depolarization. Now, here's the interesting one. We don't hear about as much. Umami. This was discovered about 1962 by the Japanese, and it is the word Japanese delicious. This is basically the detection of the meaty, brothy taste that you see, particularly with glutamine. Receptors are linked to G proteins, again, gustoductins, and they will again trigger secondary messengers inside the cell. Now, water receptors. What was I talking about there? Interestingly enough, these receptors exist in the pharynx. They respond to a volume of water intake. The sensory response is sent directly to the hypothalamus, and the response is the reduction of antidiuretic hormone, vasopressin, in response to a large drink of water. Now, it's interesting to note also that we are more sensitive to unpleasant taste than to pleasant taste. So we respond greater to bitter than to pleasant. Perhaps that is a survival mechanism to keep us from consuming stuff that would eventually lead to death. And if you think about it, even bitter tasting compounds that you find in certain plants, kids are much more sensitive to the bitter taste than adults, okay? But there are bitter compounds. We call them plant alkaloids. And some of these compounds are bitter tasting, but that also may mean that they're helpful to let us know whether or not these are supposed to be things that we are to consume or not. Another idea is taste sensitivity. Sweet and salty is overtaken by a factor of 1,000 for the sensitivity to acids. But for acids, they're overtaken by a factor of 100 to bitter. So what I'm saying is that the sense of sweet and salty, acid sense is 1,000 times more sensitive. And then when you compare acids to bitter, bitter is by a factor of 100-fold more sensitive. Okay? Now we're going to move into the eye and vision. When I say vision, this is the detection of light and the perception of visual images. And the coverage of the eye is going to include accessory structures as well as the eye itself. So if you take a look at this image here, we have to talk about the accessory features and the external structure because they play a key role in the protection and function of the eye. If you take a look at the eyelids and the superficial epithelium of the eyelids, otherwise known as palpebrae, is a continuation of skin, and its blinking keeps the surface of the eye lubricated and wiped clean of dust and grit. Basically, the eyelids consist of the palpebrae fissure, which is a gap separating the free margins of the eyelids. It's not medical, it's medial angle, the medial canthus, and the lateral angle, the lateral canthus, is the collection sites of the eyelids. Now, if you take a look at the eyelashes are basically hairs embedded in the margins of the eyelids, which prevent foreign matter from entering the eyes or the eye surface. There are large sebaceous glands associated with the eyelashes. There are also a series of modified sebaceous glands known as the tarsal glands, and they're also referred to as meibomian glands. They secrete a lipid-rich substance that prevents the eyelids from sticking together. Interestingly enough, the tarsal glands are located on the inner margin of the eyelid. The lacrimal carbuncle is a mass of soft tissue located at the medial angle of the eye that produces a thick, lipid-rich secretions, and these things kind of dry after a night's sleep. This is where you get that gritty, or sleepers, as some people will refer to them. It's interesting to note that there are muscle fibers controlling the opening and closing of the eyelid. The orbicularis oculi closes the eyelid, and the levator palpebrae superioris opens the upper eyelid. Now, the conjunctiva, if you take a look on the side, and if you take a look on the side here, you're going to see that this is an epithelium covering the inner surface of the eyelids and the outer surface of the eyeball. And it consists of a mucous membrane with specialized stratified squamous epithelium. The palpebral conjunctiva covers the inner surface of the eyelids. The bulbar conjunctiva, otherwise known as ocular conjunctiva, covers the anterior surface of the eye. Okay. What if we have bacterial infections here? Well, this is the interesting thing. Most bacterial infections are beaten off because in tears they will contain a particular substance called lysozyme, which breaks down the actual cell walls of bacteria. But it is still possible to have some infections. A stye is an infection of the sebaceous gland of the eyelash and produces a painful swelling. A chalaison is a cyst resulting from an infection in the tarsal gland. And then conjunctivitis, otherwise known as pink eye. This is an inflammation of the conjunctiva. The obvious sign is redness due to dilation of blood vessels deep in the conjunctival epithelium. Now, from here, let us move back briefly to the lacrimal apparatus, which you can see in this diagram here. We have to have a constant flow of tears to keep the conjunctival surfaces moist and clean. Tears help reduce the friction. They remove debris, they prevent bacterial infection, and they provide nutrients and oxygen to the portions of the conjunctival epithelium. The lacrimal apparatus produces, distributes, and removes tears. The fornix of the eye is a pocket created where the palpebral conjunctival becomes continuous with the bulbar conjunctival, conjunctiva. Now, in this area, the apparatus consists of the lacrimal gland and associated ducts. This is the tear gland that nestles within a depression of the frontal bone, inside the orbit and superior and lateral to the eyeball. The lateral portion of the superior fornix receives 10 to 12 ducts from the lacrimal gland. You can see that up in the B section there of the image. The lacrimal puncta are two small pores where the lacrimal lake drains. The paired lacrimal caniculi, this is where the lacrimal puncta drain into these small canals that empty into the lacrimal sac. The lacrimal sac is located within the lacrimal sulcus of the orbit. It's the site of tear drainage. At the inferior part of the lacrimal sac, the tears empty into the nasolacrimal duct. Well, that kind of makes sense when you think about it. If anybody has ever done any crying, and then afterwards you got to blow your nose because you got a lot more mucus. Well, it's not just merely mucus, it is the tears. Now, the nasolacrimal duct passes through the nasolacrimal canal, which is formed by the lacrimal bone and the maxilla, and delivers the tears into the nasal cavity. What the heck is the lacrimal lake? It's called the Lake of Tears. It's a site of buildup of tears. It's located in the medial angle of the eye. The lacrimal lake covers the lacrimal caruncle. Now, by the way, as I mentioned to you, the tears contain lysozyme. They also will contain antibodies. This is very important to attack and prevent pathogens from entry into the body. When you think about it, the eye is another area where you could have infection enter, as opposed to just thinking about the nose or the mouth. The difference with tears, though, it's slightly alkaline, will supply oxygen and nutrients. It's watery, and the nutrients are being supplied to the corneal cells by diffusion. The tarsal gland, remember we talked about this, it had sort of a lipid-rich secretion. Well, it's interesting, if you have that lipid-rich secretion covering the liquid, it helps to reduce or prevent evaporation of the watery component of the tears. Now we're going to move into the actual anatomy of the eyeball. The eyeball, of course, is roughly spheroid in shape. Orbit shape is basically shared room for both eyeball and extrinsic eye muscles. What I'm saying is that orbit space that you have in the skull, you've got to include both orbital eye fat, you've got extrinsic eye muscles that turn the eye left, right, up, down, all that other stuff, as well as the eyeball. Well, the orbital eye fat also plays a role by acting as a cushion and insulates the eyeball. Now, the eyeball will consist of 3 layers. They're also known as tunics, okay? You've got the fibrous, the vascular, the inner. The fibrous is the outermost layer, which is divided into 2 areas, the sclera, which is white, opaque, and the cornea, which is transparent, avascular, meaning no blood vessels, filled with a lot of raw nerve endings. And that's where light will go through, the cornea. In the vascular, otherwise known as intermediate layer, this is the pigmented region consisting of iris, ciliary body, choroid. And in the innermost layer, also known as the sensory layer, this is the site of the retinal cells, the neural layer, as well as pigmented cells. When you look at the eyeball also, you're talking about 2 chambers or cavities. The anterior cavity consists of the area from the cornea to the iris. This is the anterior chamber. And then from between the iris and the transparent lens, the posterior chamber. Those are both filled with a clear, watery, aqueous humor. The posterior cavity, otherwise known as the vitreous chamber, consists of the area from the posterior to the transparent lens to the retinal layer. This is filled with the gel-like substance, vitreous humor. Okay, let's go back over the fibrous layers and all the layers in detail now. The fibrous layer is the outermost layer of the eyeball. It helps to support and protect the eye. It's rich in collagen fibers. It's also going to serve as the attachment site for the extrinsic muscles and contains structures that assist in focusing the image. The sclera consists of a dense fibrous connective tissue that contains both collagen and elastic fibers. The layer is thickest over the posterior surface of the eye, near the exit of the optic nerve. The extrinsic muscles attach to this layer, and the layer prevents light from passing through the sclera to affect the retina. The major point I was trying to say, and I may have said this in lecture, is this. You only have a limited area where light can come through. That's going to be the cornea, initially. The sclera keeps the rest of the eye from being stimulated from other sites where light might pass through. The cornea is a structure continuous with the sclera at a boundary site called the corneoscleral junction, also known as the corneal limbus. This consists of a dense matrix of multiple layers of collagen fibers, but there's no blood vessels, lots of raw nerve endings, which makes it very, very sensitive structure, as well as superficial epithelial layers get oxygen and nutrients from tears, and internally, where do they get the nutrients and oxygen? From the aqueous humor. Now, the vascular layer, let's talk about that for a minute, otherwise known as the uvea. This is a pigmented region that includes the iris, the ciliary body, and the choroid. Contains numerous blood vessels, lymphatic vessels, and intrinsic, that is, inner smooth muscles of the eye, especially around the iris and ciliary body. Its function is to provide a route for blood vessels and lymphatics that supply the tissue of the eye, regulates the amount of light entering the eye, secretes and reabsorbs the aqueous humor that circulates in the chambers, especially of the anterior cavity, and we'll go over the patterns of circulation shortly. And then it also controls the shape of the eye, so it's essential for focusing. We also have the structures, the iris, the ciliary body, and the choroid that we have to go into a little bit more in depth. The iris is a flattened ring structure containing blood vessels, melanocytes, and 2 layers of smooth muscle. These are the pupillary muscles. The dilator pupillae extends radially from the pupil edge and causes the pupil to dilate, usually due to sympathetic stimulation. And the sphincter pupillae, which is located on the edge of the pupil, causes the pupil to constrict, usually due to parasympathetic stimulation. So the pupil size controls the amount of light entering the eye. Now the ciliary body is a thickened region. Okay, I just want to make sure we get to the site where we have, there we go. We'll just go for that right now. The ciliary body is a thickened region deep to the corneo-scleral junction. The iris attaches to the anterior part of the ciliary body. The posterior part extends to the level of the ora serrata. This is the serrated anterior edge of the neural layer of the retina. Okay? The bulk of the structure consists of the ciliary muscle, the ring of smooth muscle that extends into the interior of the eye. Now, this muscle is covered with a wrinkled epithelium covering called the ciliary processes. Now, as you can see there, the ciliary processes also have some very interesting little fibers that attach to them, which then attach to the lens. Okay? This is the ciliary zonule, which we're going to get into in a few minutes. Okay? These suspensory ligaments, otherwise known as the ciliary zonule, attach that inner lens, and they're going to pull the lens to alter the shape, and hence the magnification power of the lens. Now, the choroid is the vascular layer that separates the fibrous layer and the inner layer posterior to the zona serrata. The choroid layer is covered by the sclera and then covered by the retinal layer. The vast capillary network of the choroid provides oxygen and nutrients to the retina. You should also know that the choroid layer contains melanocytes for pigmentation, which happen to be very rich on the layer near the sclera. Okay, and you can see this a little bit here. Now what we're going to do is go back for a minute and talk about the retina. That's the innermost layer. And it consists of a thin pigmented layer and a thicker neural layer. Now, the pigmented layer contains pigmented cells that support functions of the photoreceptors of the retina, as well as absorb light that passes through the neural layer to prevent a bounce back of photons back to the photoreceptors and hence create visual echoes. The issue here is that once the light passes through the retina, it's not going to be reflected back. It's going to be absorbed, in essence, by that pigmented layer. The neural layer consists of the photoreceptors. Rods for light, they're sensitive to even low light, such as moonlight, but lack sensitivity for color. The cones are sensitive only to higher light intensities at selective frequencies. They have cones for red, green, blue, and they also provide visual acuity of objects. Now, if you sit there and say red, blue, green, okay, that's cute, but wait a minute, we got other colors. Yes, you're going to go over that in a few minutes about how they have different types of overlap. Now, there's a third type of photoreceptor called the intrinsically photosensitive retinal ganglion cell, IPRCG. Contains a different photopigment. It's called melanoopsin. Now, these cells respond to different light levels and influence the body's 24-hour circadian rhythm, the biological clock. That information is going to get passed over to several other sections of the brain to help it coordinate on timing. Okay, when's daylight, when's night. Now, the distribution of rods and cones is not uniform. Rod density declines as the image moves from the periphery of the retina toward the center. The cone's density is very concentrated around the central area, where visual image arrives after passage from through the cornea and then through the lens. This central site of the image is called the macula. Okay? Now, the cones are highly dense there. There's actually no rods. The center of the macula is called the fovea centralis, or fovea, and is the site of the sharpest color vision. Now, if you remember, I told you that basically color requires high light. If you're trying to look at something under something like moonlight, all you're going to see are differing shades of whites and grays, etc. If you want to see something in very, very dim light, don't look at it straight on, because obviously it wouldn't be getting a good image at that fovea centralis, but look at it from sort of at an angle off of the center, and that's where it'll be a little bit more acute in the vision, because literally you're going to be looking at using your rods as well. Now, we come back to this concept called the visual axis. This is the imaginary line drawn from the center of the lens to the fovea centralis. This helps us to comprehend the image formation on the back of the eye. So we have to keep in mind that the eye has different layers, or the retina has different layers into it. And here's what we have. Rods and cones. It's interesting to note they face in the opposite direction, away from the light. Bipolar cells, which are the neural cells that the rods and cones synapse with. Ganglion cells, with which the bipolar cells synapse with and carry processed image signals away from the retina via the axons of these cells. And you can see this where all of the axons come together at the optic disc, which we'll get into in a minute. But then we've got this other interesting thing. We've got these other cells there called the horizontal and amacrine cells. Horizontal cells is a network layer of nerve cells between the photoreceptors and the bipolar cells. The amacrine cells are a network layer of neural cells between the bipolar cells and the ganglion cells. What these cells facilitate or inhibit is the communication between each layer of the cell, between rods and cones, bipolar cells, ganglion. And what they play a role also doing is modulating the sensitivity of the retina in variations of light sensitivity. Finally, we come back to that point I was talking about a minute ago, which was the optic disc. That's located just medial to the fovea centralis. This is the site where the ganglion axons have penetrated outside of the eyeball. In essence, all of those axons are going to help form the optic nerve. Also, the central retinal artery and vein pass through, sort of in the center of this optic nerve, and they come out around the area of the optic disc. This is going to provide blood to the choroid. Okay? Now, you got to keep in mind one other thing. Since rods and cones do not exist there, this site has also been referred to as the blind spot. If you want, take a look at figure 17.8 for the demonstration tool. Focus one eye on the particular dot, start moving away from the page, and you'll notice that the plus symbol disappears because the image of the plus symbol has moved over to the actual optic disc. There's no rods or cones there. There's no perception of that image, but continue to keep moving back out, and boom, it shows up again. Now, let me move back to the chambers of the eye for a few minutes here. The eye is divided into two cavities, anterior, posterior. The posterior cavity, otherwise known as the vitreous chamber, contains this gel-like mass called vitreous humor. Now, you have to understand that this gel-like material, which consists of specialized cells that produce collagen fibers and proteoglycans, this jelly-like material helps stabilize the shape of the eyeball as well as help maintain the retina by holding it up against the choroid layer. If the vitreous chamber, if the vitreous humor were to leak out, you would have the collapse of the retina away from the choroid. Now, the challenge also is this: the vitreous humor is formed during development. It's not replaced. So loss of it means that you won't have regeneration the way that you would have, let's say, a cut or something else. The anterior cavity produces aqueous humor, which is sort of a clear fluid. It's similar to cerebral spinal fluid in composition. And now its circulation starts off from really the ciliary process, where it's made and then it circulates around and up over by the lens, and then up through the pupil, and then up over the body of the iris. Now, this is made by an active secretion of the epithelial cells of the ciliary body process, which I've mentioned. And this basically, the process cells will control the composition. This aqueous humor does play a role in nutrient and oxygen delivery as well as waste removal. It helps maintain the shape of the eyeball. It helps account for the pressure of the eyeball also, the intraocular pressure. The normal range of intraocular pressure is 12 to 21 millimeters of mercury. Now, basically, you're producing, on average, 1 to 2 milliliters per minute. Reabsorption is going to occur at the scleral venous sinus, otherwise known as the canal of Schlemm. This is located in the corneoscleral junction. This delivers the blood into the scleral veins. Now, if you notice our lens is biconvex, outwardly curving, flexible disc, consisting of concentric layers of cells and covered by dense fibrous capsule. The disc contains many elastic fibers which normally orient the lens in a spherical shape. Okay. Around the edges of the lens, these fibers intermingle with the ciliary zonal fibers, upon which pulling will flatten the shape of the lens. So if they pull tight, the lens sort of flattens out and you lose some of the magnification power, whereas if you allow it to fully just relax, it will have a greater magnification power. Usually what happens is you will have accommodation, so you will have these changes in the shape. But also the thing that's interesting about this is that the interior of the lens, there are structures called lens fibers. These are cells that have lost their nuclei and most other organelles. They're long, they're slender, they're filled with transparent proteins called crystallins. Crystallins provide the focusing power and clarity of the lens. Now, these proteins usually remain stable, intact, and functional for the entire lifetime. There are situations such as exposure to ultraviolet rays, gamma radiation, infrared radiation, reactions of drugs that can help to cause basically the denaturing of the proteins, which then means you have a loss of transparency and leads to the formation of a cataract. Now, we know today in modern medicine that we can actually remove the lens later on in life and put in an artificial one, which then restores some vision and visual clarity for the patient. Now, we're going to move from there to vision concepts and get into actually issues of photoreception, etc. I do encourage you to review the spotlights on refractive problems and photoreception. As we said before, rods detect light, cones detect color. The visible spectrum wavelengths are basically from 400 to 700 nanometers. That's the red, orange, yellow, green, blue, indigo, and violet. That's the parts of the spectrum. You need to know that there is some overlap to the sensitivity of each of the red, green, blue cones to be responsive to the other colors. You can see where the rods are responsive. You can see where green cones are, red cones are, and blue cones. Now, one of the interesting things, if we go back a minute, is that light pathways get bent, that is refracted through different mediums, whether it's glass, whether it's liquid, water, or whether it's the gel, the vitreous humor, etc. Much more so if you have structures such as lenses, okay? What will happen is you can have them all come to a certain point as these light rays focus back on a particular point. That's known as the focal point. That's a specific point of intersection on the retina of all the image points. The focal distance is the distance between the center of the lens and its focal point. The focal distance is determined by two factors: distance of the object to the lens, shape of the lens. Okay, the curvature of any lens influences the focal distance because light rays may be bent and can be refocused once they pass through the lens. Okay, you got to keep your mind on this idea. The lens, in part, inside your eye, is properly set up to focus on an image on the back of the retina. And this is where we get into problems. You want to have close vision, you want to have really intense vision, you're going to have a curved lens. You want to have distance vision, you have to have a more relaxed lens, almost flattened out, so that the focal point will still be on the fovea centralis in both cases. And you can see how you have a reversal of images because of lenses also. Now, but I don't see it that way. I see a telephone pole and it's still sticking up. That's because your brain does the process of reversing the image so that it appears properly in its proper orientation by the time it reaches the brain. Okay? And by the way, if we have any distortions on the cornea, well, the cornea doesn't do a lot of magnification, but if there are thickenings or waves or roughness on the cornea itself, these visual distortions will lead to astigmatism, so the visual image will be distorted or blurry. The concept of accommodation, that's the automatic adjustment of the eye to obtain a clear vision. This is basically an interaction between the brain, the occipital lobe, the ciliary body muscles to alter the shape of the lens by pulling on the lens via the ciliary zonule. Now, visual acuity. You know, when you sit there and say, well, this person has 20/20 vision, what are we really talking about? We have really a comparison of the normal standard. Standard vision rating of 20/20 means that the image detail of viewing the object at 20 feet from the viewer is the same with the person's vision. So in other words, what I'm saying to you is a normal vision person will see the letters, the blocks, whatever you want, 20 feet away, and they can comfortably see that without any adjustments, without any squinting or anything. If a person had 20/30, they have the capability of viewing the detail at 20 feet that a person standing 30 feet away would have. If someone has a visual acuity below 2200, what I mean by that is 2300, 2400, even with the help of contacts or glasses, the person is still considered legally blind. If you have to think about it this way. A person has 2200 vision. They only have the visual acuity that you would see at 20 feet when they're 200 feet, when a normal person is 200 feet away. In other words, they can see detail only if you compare them to a normal person. The normal person would be able to have the visual acuity of 200 feet away. Obviously, that's really bad for an individual who's 20 feet away from an object but can't see unless they have huge letters, huge objects, etc. This is where we get into these concepts or terms. Emmetropia, normal vision. That's a normal focusing of the image on the retina surface. Myopia, that's nearsightedness. Due to the lens shape and eyeball shape, the focused image is projected in front of the retina. Therefore, correction must be done with diverging concave lenses to put the image back against the proper area of the retina. Hyperopia is farsightedness, and this is due to the lens shape or eyeball shape. Again, the focused image is projected behind the retina surface. Corrective lenses are done using converging lenses there. Now we have another unusual concept that does pop up, particularly for adults after age 40. It's called presbyopia. This is where the lens loses some elasticity, and hence minor corrective lenses are necessary to achieve close-range vision. Okay? Total blindness, when we use that term, implies total absence of vision. This can be due to damage to the eyes or the optic pathways. And the causes include diabetes mellitus, cataracts, corneal scarring, retinal detachment, accidental injuries, or hereditary factors. Let's talk about the photoreceptors for a few minutes, shall we? Now, if you take a look at this image, both cones and rods contain visual pigments. These are organic compounds that alter shape due to exposure of light and act in the detection of light for the photoreceptors. They're located in each of the photoreceptors' outer segment in flattened membrane structures called discs. So it is the discs that are the actual immediate detectors. Notice that the cones and the rods are pointing toward the pigmented surface. Light is coming up from behind them. Okay, that's important to keep in mind. The discs reside in the outer segment, Whereas the inner segment of these photoreceptors has the most of the cell's organelles. We're talking about nucleus, mitochondria, Golgi apparatus, and then releases its neurotransmitters. As you can see, the back part of that, the photoreceptor synapses with the bipolar cell. Visual pigments are derivatives of rhodopsin, which is found in cones and rods. Rhodopsin consists of a protein, opsin, attached to a pigment, retinal, and retinal is synthesized from vitamin A. All rods have the same form of opsin. Cones have the same retinal, but different opsin that respond to the different wavelengths of light. So they have to respond to the different wavelength ranges, for both red, green, and blue. When you take a look at somebody's normal vision, the cone population consists of 74% red cones, 10% green, 16% blue. Then we get into the other issue of photobleaching. And I'm going to basically bring this down to some simplistic areas and hold off on resting state, dark vision, a dark view, and all that stuff. Here's the main point. Once a photon, a light particle, hits the rhodopsin molecule, the retinal part shifts and leaves the opsin particle in the basically becomes activated. The retinal part shifts its shape and leaves the opsin part of the molecule, and the breakdown of rhodopsin is referred to as bleaching. Okay, bleaching. Now, a new retinal molecule can be inserted into opsin with the help of an ATP-dependent enzyme reaction. The opsin activation leads to the activation of a G protein transductant, which leads to the activation of the enzyme phosphodiesterase, then leads to the reduction of the sodium ion flow into the cytosol and eventually reduction of the neurotransmitter. So in essence, the action causes the bipolar cells to signal that a photon was absorbed by the rod or cone, and that signals the stimuli to begin the process of visual stimulation. See, in other words, the neurotransmitter release is kept basically going until you have sodium coming in. Sodium coming in is because basically the photon was absorbed, changed the shape of the retinal from a angled part to a straight part. Opsin kicks it out. You have to have a new retinal enter it for it to be functional again. Okay? Once there's a change in the amount of neurotransmitter release, the bipolar cell detects it and then signals the ganglion cell. We had a photon absorbed. Okay? Now, let's go from here to visual pathways for a few minutes. From the retina, the visual stimuli is passed along the optic nerve. This is cranial nerve number 2 until it reaches the optic chiasm. Now, this is where you get into a little bit of what the chiasm is for. You have the division of the left field and right field of each eye, so that the left field of both eyes passes down one optic tract. The right field of both eyes passes down the other optic tract. That's where the colors are involved there, if you can see that. The optic chiasm does that division. From the optic tracts to the visual information will pass into the lateral geniculate body of the thalamus. Now, some of the connections pass to the superior colliculi. This is for visual reflex coordination. You will also get information passed to the suprachiasmatic nuclei of the hypothalamus for circadian rhythm timing. And then the projection of the fibers goes to the also to the diencephalon for the pineal gland and to the brainstem. But basically from the thalamus, the projection fibers, that is otherwise known as optic radiation, project into the visual cortex of the cerebral hemisphere, basically the occipital lobe. And this is where you're going to get further visual processing. I hope that's helpful there. Now let's move into the equilibrium and hearing. What you want to keep in mind is that equilibrium is a sensation that provides position of head in space via monitoring gravity, linear acceleration, and rotation. In other words, all the movements you do Whether you're the fancy skater doing the triple spins, or whether you're just basically riding your bicycle or getting up every morning, you have to have some sense of the orientation and position. Where is gravity? Where is bottom? Where is up? Where is side to side? Hearing, though, is the sense to detect and interpret sound waves. Now, the process requires transduction. Transduction is basically changing from one energy force to another. In this case, you're going to have sound waves, two mechanoreceptors, the hair cells, that convert information into action potentials. Equilibrium is obtained from gravity, linear acceleration, rotation motions, to mechanoreceptors, the hair cells, that convert the information into action potentials. The equilibrium info is sent to the brainstem, and the auditory info is sent to the auditory cortex. We'll get more into that in a few minutes. Let's talk first about the anatomy of the ear. The ear is divided into external, middle, and internal. The external we're familiar with, that's that fleshy cartilage structure, the pinna, and the external auditory canal. It ends at the tympanic membrane, the eardrum. The pinna helps to collect sound waves. The ear canal has ceruminous glands, which make earwax, serum, and basically that's used to inhibit microbial growth, and also hairs that will inhibit debris and insect infiltration. The middle ear starts from the tympanic cavity that includes the auditory ossicles. Now, you've got the tympanic membrane there, and the sound vibrations are transferred via mechanical vibrations to the tympanic membrane, then to the malleus, which is attached to the membrane, then to the incus, then to the stapes, which is attached to the oval window of the inner ear. Round window. I want to make a correction there. The middle ear is also an air-filled chamber with and basically you have this connection with the auditory tube. Now, it's also referred to as the eustachian tube or the pharyngotympanic tube. Basically, this tube helps equalize the air from the outside with the middle ear chamber. The chamber also has two of the smallest skeletal muscles, which are attached to the auditory ossicles. These are useful in helping to reduce exposure to very loud noises. That's the tensor tympani. Now, these are both skeletal muscle type of muscles, and they're innervated by the mandibular nerve V3 of the trigeminal nerve. And basically, the tensor tympani is attached to the malleus. The stapedius is innervated by the facial nerve, which is cranial nerve number 7, which is attached to the stapes. Okay. Now, what about the internal ear? This is surrounded by the petraeus, that is the bony part of the temporal bone. And it has sensory processes for both equilibrium and auditory cochlea. And it consists of the labyrinth, which is a network of canals. The contour of the structure is surrounded by bony labyrinth, then inside of it is followed by membranous labyrinth, which is a fluid-filled tubular structure. The space between the bony and membranous labyrinth is filled with perilymph fluid, whereas the space inside the membranous labyrinth is filled with endolymph fluid. Now, I want you to understand that the perilymph is similar in composition to cerebral spinal fluid, but endolymph has a much higher content of select electrolytes. If you go to your textbook, into the appendix on page A1, there will be an appropriate comparison of CSF, of endolymph, and of perilymph. So I encourage you just to take a perusal at that. Also, the receptors of the internal ear are found within these tubes. So let's talk about this for a few minutes, the internal ear further. And what we're going to talk about is that the structure is subdivided into the vestibule and the semicircular canals. And basically in the vestibule, this is a pair of membranous sacs, the saccule and utricle, which provide position and acceleration movement info, basically response to gravity. Semicircular canals provide rotational movement info. Basically, they do this in the 3 dimensions, X, Y, and Z, okay? They're also known as the semicircular ducts. Now, this together is referred to as the vestibular complex. The fluid-filled chambers are continuous with semicircular canals. When we get to the cochlea, though, that's a spiral-shaped, bony chamber that contains the cochlear duct. Receptors provide the sense of hearing. The duct is placed between a pair of paralymph-filled chambers and has the stapes attached to the oval window, whereas the pressure displacement is relieved by the round window. Did I get that mixed up? And I sit there and I want to make sure. But basically, what you have, think about it as if it was a snail shell, but if you could unwind that whole snail shell, what you'd see is a middle chamber filled with endolymph, surrounded by a larger chamber, actually 2 chambers sort of, and they're filled with paralymph. Now, equilibrium. The receptors of the vestibular complex are the hair cells. Each receptor hair cell contains specialized cilium called kinocilium, which is the largest, and stereocilia. Now, these cilia basically are embedded into this larger structure, the ampullary cupola. It's a jelly-like material. But let me help you to understand this. The kinocilium is the largest. The stereocilia is basically somewhere in the range of 80 to 100 are present. The hair cells are then attached to a sensory nerve, end, which is upon which the hair cell stimulates the neuron cell to fire action potentials. Now, the semicircular ducts are divided into anterior, posterior, and lateral semicircular ducts. This is continuous with the utricle. Each duct contains an ampulla, expanded regions where the hair cells reside, surrounded by endolymph. Now, in the ampulla, the hair cells, otherwise known as the ampullary crest, emerge from the wall of the ampulla, and they're embedded into this gelatinous structure, the ampullary cupola. Okay, so you can see this from the diagram, these hair cells embedded into it. Now, how does this really work? When forces of gravity shift, the direction of the flow of the endolymph pushes in one direction or the other. And what happens is that this tugs on the hair cilia, and that results in the altering of the firing of the hair cell to the sensory cell, to the sensory neuron, and therefore by it alters action potentials. Since both sides of the head have semicircular ducts, these firings are different and processed by the brain from the respective left-right semicircular ducts of motion and rotational movements. So keep in mind, you have one set on one side, one set on the other, and basically you're going to get a differing amount if you spin around or if you jump up and down, etc. Now, let's get into the utricle and the saccule. Oh, by the way, here's a great picture of the hair cell. And as you can see, most of the hair cell, a lot of organelles and such around it, but then you have that kinocilium, and then you have the stereocilia, and they're just basically embedded into the gelatinous material. And then intimately wrapped around the hair cell is the sensory nerve ending, and of course you've got some supportive cells there too. But basically, once you start having this pull in one direction or the other, it's going to stimulate or inhibit the hair cell. That information is going to be passed on to the sensory nerve ending and then to the rest of the brain. In the utricle and the saccule, each site is connected by a narrow passageway called the endolymphatic duct. Both utricle and saccule are similar in that you have the chamber has hair cells. These are clustered on oval structures called maculae. All right? And you have also endolymph. The utricle is sensitive to changes in the horizontal movement. The saccule is sensitive to changes in the vertical movement. The hair cells processes, the cilia, are embedded into a gelatinous structure called the otolithic membrane, and you can see that here. But here's another pointer. This is not just a blob of gelatinous material. On top of it is something that adds further mass to it and enhances the sensitivity to changes in the upright position, downright position, lateral positions, etc. And it is these dense layer of calcium carbonate crystals called ootliths, otherwise known as ear stones, that really give it some mass, so that if you move forward, you move backwards, you move around, you dropped, the head moves posturally or anteriorly, gravity will cause the ootliths to shift downward or upward, respectively. These hair cells that are embedded into the gelatinous mass are going to get pulled in one direction or another, and you're going to get a resultant change in the sensory nerves firing APs, action potentials. Okay? Now, from here, we got to talk about the neural pathways to equilibrium sensations. Hair cells firing leads to sensory neurons firing their APs. Now, this is all located in the vestibular ganglia, which then extends its axons into the vestibular nerve, which then merges into the vestibular cochlear nerve. Yes, another cranial nerve, number 8. The vestibular cochlear nerve innervates the pair of vestibular nuclei located in the boundary of the pons and the medulla. You can see that there, the vestibular nucleus. This information is then relayed to the following: to the cerebellum. Remember, we got to have that coordinating, etc. To the superior colliculi, which basically this is involved with eye motion control and allowing fixation of eyes on a target despite its movement or motion. To the cerebrum, this is going to provide us with the conscious awareness of motion. And then to the cranial nerves involved with eye, neck, and head movement, and that's cranial nerves 3, 4, 6, and 11. Okay? Now, from here, let's talk about hearing. Hearing is the perception of sound. Sound waves consist of waves of pressure conducted through the medium such as air. In this case, you're going to have the air molecules having areas of high density and those that are going to have areas of low density. And basically, they travel in what we would call waves. Waves have a height factor that is called amplitude, and they have a frequency concept that is basically cycles per second or waves per second. And another way to say the frequency is hertz. Okay? So wavelength is determined by the number of waves that pass per second of time. The frequency of sound is the number of waves that pass a fixed reference point in a given time. Physicists, and you're going to see this when it says cycles, they use cycles instead of calling it waves. Now, we measure the frequency of sound in cycles per second, the hertz. Our sensitivity, human beings, to sound ranges from 20 hertz to 20,000 hertz. Keep in mind, dogs have a much higher amount, maybe in the 30s. Mice have even a higher amount, etc. Anything above our hearing range is referred to as ultrasound. Anything below the lower end range, such as 19, 17, 18, 16 hertz, that's referred to as infrasound. Now, wavelength and frequency are inversely related. The greater the frequency, the smaller the wavelength. We measure sound energy in decibels. If you take a look at this intensity of representative sounds, this is critically important because what happens is this is what determines, in some cases, whether people get injured over a period of time working in factories and noisy places, etc., or whether they truly do need hearing protection, etc., and also the range in which they're able to function. Okay? The greater the amount of energy in a sound wave, the intensity determines how loud it is. Now let's talk about the anatomy of the cochlear duct. This consists of 3 ducts or 3 scalas. The cochlear duct, the scala media, is filled with endolymph and is sandwiched between a pair of perilymph ducts, the scala vestibuli, the vestibular duct, and the scala tympani, the tympanic duct. All 3 are encased in a bony labyrinth, except for the round window at the base of the scala tympani and the oval window at the base of the scala vestibuli. Okay? The scala tympani and the scala vestibuli are interconnected at the tip of the cochlea. Think of the cochlea as like a long tube, and you rolled it and rolled it and rolled it up until it appears sort of like a snail shell. That's where they get the cochlear term for it. The hair cells of the cochlear duct are in a structure called the spiral organ, otherwise known as the organ of Corti. This structure rests on a basilar membrane which separates the cochlear duct from the scala tympani. And you can see that here. The hair cells lack kinocilia, and the stereocilia, interestingly enough, are arranged in a series of longitudinal rows. They're embedded in an overlying tectoral membrane. Tectum refers to roof. The tectoral membrane is firmly attached to the upper wall of the cochlear duct. So once the hair cells, stereocilia, are pulled, the hair cells will fire and stimulate the sensory neurons beneath, as you can see here. So what happens is that these cells, the hair cells, both the outer set and the inner set, basically have rows of hair cells. When someone becomes deaf from overexposure to sound over a long period of time, they will see what basically is hair cells that they use electron microscopy that are all broken down and they're all in disarray. They're definitely not attached to the tectoral membrane. Let's go through the physiology of hearing in 6 basic steps. This is a perfect example of transduction. The sound waves arrive at the tympanic membrane. The movement of the tympanic membrane displaces vibrations of the auditory ossicles. The movement progresses from the malleus to the incus to the stapes. The stapes is attached to the oval window. Now I say this and I get this mixed up, but I'll clarify this basically in that it is the oval window and that the pressure distortion comes out the round window. Movement of the stapes at the oval window produces pressure waves in the perilymph of the scala vestibuli. The pressure waves distort the basal membrane all on their way to the round window of the scala tympani. The vibration of the basilar membrane causes the vibration of the hair cells against the tectoral membrane. Information about the region and intensity of the stimulation is relayed to the CNS over the cochlear nerve. What you have to understand is this. The entire link to the basilar membrane becomes a translation tool. Anything that I say to you is probably noises or sounds that are in a variety of hertz. So, the closer to the oval window for the basilar membrane is sensitive to much higher frequencies of sound than further away, as you can see there, 1600 hertz close to the oval window, 6000 about mid-range, and 1000 hertz much farther close to the tip of the entire cochlear structure. Okay? And so this is how we discriminate in various sounds, because at the same time you may have several different parts of the basilar membrane having distortions and then firing signals that say this sound has a complex mix of frequencies. Okay? Now, since we've gone through all of that, let's talk about the auditory pathways. Stimulation of the hair cells at a specific location of the basilar membrane activated those sensory neurons. Yep, we know that. Sensory neurons carry auditory information in the cochlear nerve. The cochlear nerve and the vestibular nerve merge together to form the cranial nerve 8, vestibulocochlear nerve. The information from the cranial nerve number 8 enters the medulla to synapse at the cochlear nucleus on each respective side of the head. Remember, you've got one on each side of the head because we have ears on each side of the head. This information will ascend to both the superior obligatory nuclei of the pons and to both inferior colliculi of the midbrain. Remember, inferior colliculi is involved with auditory reflexes. Okay? The midbrain processes the unconscious motor responses, particularly when you have sudden or loud noises. Okay? And you can hear this. You hear some bang and people turn their head toward it without even thinking. Then you have the ascending auditory information progress to the medial geniculate body of the thalamus and then passes on to the auditory cortex of the temporal lobe. Whereas you can see there is a range of processing that occurs depending on the low frequency, high frequency, as it's going through parts of the auditory cortex of the temporal lobe. Well, that's just about it. I encourage you to review the chapter review, peruse the review questions, review the clinical terms. Again, remember that's fair game for any exam or quiz. And then also review the spotlights on olfactory ingustation, refractive problems, and photoreception. And you can now begin to prepare for lecture exam number 3.